Good evening, everyone. Hope my voice is audible. Yes. Good evening, everyone. Yes. So my slides are also visible. Mhm. So for today's session, I have planned to take recombinant DNA technology, right? So what is recombinant DNA technology and what are the objectives? Objectives of the session would be you would be knowing what is the definition of recombinant DNA technology, the tools that you use for recombinant DNA technology, and procedure for the recombinant DNA technology and steps of recombinant DNA technology, and finally the applications where all we use this recombinant DNA technology. Now, starting with the definition. So what is this recombinant DNA technology? This is a method of genetic engineering. Why is it a method of genetic engineering? Because you are engineering your genes. Yes, you're working with your genes or you're working with your DNA. So for how do you do that? You do that by creating a recombinant DNA. So what is a recombinant DNA? Recombinant DNA is such that where you introduce a gene from one organism to another organism. That is what is a recombinant DNA. This is not naturally occurring. Yes, you are introducing one gene from one organism to the vector of another organism. So that is what is recombinant DNA. By utilizing this, if you are going to amplify your gene of interest, then that kind of a technique is called your recombinant DNA technology. Right? So it is obtained when genetic material from one organism is artificially introduced into the genome of another organism. Right? So first of all, if you have to understand what I meant by this amplification of DNA. So how do you do this amplification of DNA? For which you have to know about the term cloning. What is cloning? Cloning is a technique by which you produce multiple copies of your DNA, right? So you produce multiple copies of your DNA. So cloning is again divided into either cell-based cloning or enzyme-based cloning, right? So what is the cell-based cloning? That is, you produce this recombinant DNA, you introduce it into another cell, yes, wherein you allow it to multiply. That is what is called your cell-based cloning. So here in recombinant DNA technology, we are using the cell-based techniques. That is, we'll produce a recombinant DNA, we'll introduce it into the host cell, and then we will allow it to multiply inside the host cell. That is what is cell-based technique, and that is a in vivo technique because we are utilizing the cells machineries. Yes, we are utilizing the cells protein synthesizing machinery to produce multiple copies of our DNA of interest, right? Whereas what is in vitro? If I take a test tube and I put inside the my DNA of interest and the enzymes that are necessary and the buffer that is necessary, all the nucleotides that is necessary to produce multiple copies. I give all the essential things, all the essential ingredients for the synthesis of DNA inside a test tube, and then I allow it to multiply. I'm providing the environment for it to multiply. Then that kind of a technique is called as the in vitro amplification of DNA, and that is what we use in a PCR or polymerase chain reaction. Now, in this session, we are going to focus on the recombinant DNA technology. Now, what are the steps? So in gist, I will tell you the steps. One is the isolation of DNA of interest and the vector DNA. So what should you do? I want a DNA of interest. I want to amplify one DNA of interest. So I'm going to isolate the DNA of interest. Mostly it would be the human DNA of interest, right? So human gene of interest, I'm going to isolate. And also I have to take the vector DNA All right. Now I'm going to cut both of these with the same restriction endonuclease. So let us come to the tools of the DNA recombinant DNA technology in the further slide. But for now, you understand that you have to cut it with restriction enzymes. So I have the DNA of interest. I have the vector, which I usually use a plasmid. Right. So this I'm going to subject it to a restriction endonuclease, wherein I get fragments of gene of interest, and then the vector also gets cut at specific site. All right. Now I'm forming a recombinant DNA in which I have the vector with the gene of interest. Now I will introduce it into a host cell. Yes, I will introduce it into a host cell, and then it will allow it to multiply. So I will get protein products out of it. So I get so many multiple copies of this gene of interest as and when the cell replicates or as and when the cell divides. And then I will get multiple products out of which I will get the protein product also. This is the overview of the steps in a recombinant DNA technology. Now, talking about the tools of recombinant DNA technology. So tools, what have I told you in the steps while telling you the overview of steps? I told you there is an enzyme that is necessary, that is the restriction endonuclease, right? So the restriction endonuclease is one main requirement. And also I have the vectors. Yes, I have the vectors and I have the host cell. I need a host cell, right? I need a host cell and various transfer techniques. Right, I need various transfer techniques. So first thing is we will see each of these one by one. The first and foremost requirement will be your restriction endonuclease. Yes, so this is what we had seen already. So I will tell you about restriction endonuclease, after which we will again come to the overview, right? So the restriction endonuclease is, what is it called? It is called as the molecular scissors. What is it called? It is called as the molecular scissors. Why is it called as a scissor? Scissor means it is going to cut something, right? So this is the endonuclease. Yes, what is an endonuclease? Endonuclease is an enzyme that will cut your DNA in between. Yes, what is on the other hand, in contrast, what do you call an exonuclease? Exonuclease is something which cuts at the ends of your DNA. Yes, whereas endonuclease, somewhere in between your DNA at any specific site it will cut. That is what is the endonuclease. Now that is why it is called as a molecular scissors, right? So how have we identified it? So from where have you identified this restriction endonuclease? So we have identified it from it is also known as, what is it? It is also known as molecular scissors because it is cutting my DNA. Right? And then how did we discover it? How did we discover this restriction endonuclease? Where is it present? It is present in the bacterial cells. So in the bacterial cells, it is used as a defense mechanism. Yes. So whenever a bacteriophage infects a bacteria, what will it do? It will try to get inside the bacteria, right? So whenever the phage infects the bacteria, it will try to get inside the bacterial cell. So once the gene of this bacteriophage enters the bacteria gets produced, starts producing restriction endonuclease that will go and cut the phage DNA and protects itself, right? So that is considered as a defense mechanism. So we have so many restriction endonuclease which are isolated from many bacterial cells, right? But the host DNA shouldn't be affected, is it not? So in the bacteria, there will be the host DNA, bacterial DNA. You don't want the bacterial DNA to get affected by the restriction endonuclease, for which this bacterial DNA will get methylated. Yes, so that restriction endonuclease will only attack the phage DNA and not your methylated DNA, right? So this is how it was discovered. And it cuts at specific sites. Yes, how does it cut? Where does it cut? What is this specific site? This specific site is called as the palindromic sequences. Yes, this specific site is called as the palindromic sequences. What is a palindrome? Palindrome, for example, I will tell you, suppose there is this word madam. If you read from right to left or from left to right, it is going to read the same. Is it not? That is called as a palindrome. So palindromic sequences means you have this, suppose you take this example. If you read the sequence from 5' to 3' in one strand and the 5 prime to 3 prime in the other strand, then it is going to read the same. That is what is a palindromic sequence. Now, along this sequence only, your restriction endonuclease cuts. And each of the restriction endonuclease will have specific sites for itself, right? So this is all about your restriction endonuclease, right? So it from where have we identified it? We have identified it from the bacteria. So many bacteria produce different restriction endonucleases. They use it for their defense mechanism to protect itself from the bacteriophage, right? And they cut in the specific sites called the palindromic sequences, right? So now let me tell you, there are two types of cleavages. So one is the restriction endonuclease, it cuts, is it not? So how does it cut? So two types of cuts are there. One is sticky end producing restriction endonuclease, and the other one is the blunt end producing restriction endonuclease. So depending on the type of the restriction enzymes and the recognition sequences, the pattern of cutting will be different in different sequences. So thereby it produces sticky ends or blunt ends. So what do you mean by sticky ends? Yes, what do you mean by sticky ends? Suppose you take this example. Sticky ends means, I have this example 5 prime GAATTC 3 prime. Yes. So again, I have this palindromic sequence on the other strand also. So how does this cut? How does my restriction endonuclease cut? It cuts like this. Okay. So it cuts at different places. It doesn't cut straight away. It cuts at different places in the two sequences. Yes, it cuts at different spaces and two different strands of your double-stranded DNA. So in that case, look at the product that we have got. Yes, this is a product that we have got. And there is the hanging ends which are present because of this kind of a uneven cut, right? So those ends are called the staggered ends, yes, or the hanging ends, and those are sticky ends. Why do you call them as sticky ends? You call them as sticky ends because as you can see, they are complement to each other, right? So according to Watson and Crick base pairing, it will just reunite itself. So it can just anneal itself, right? So these kind of cuts or sticky ends, because they are the sequences of the hanging ends are complementary to each other, and they can come closer and get annealed. So that is what is the speciality of sticky ends. Whereas the other type of ends is the other type of restriction endonuclease is that one which produces the blunt ends. Blunt ends means I'm cutting the restriction endonuclease cuts straightly along the two strands of the DNA. So as it cuts, it produces blunt ends. Yes, as it cuts, it produces blunt ends. So this is what is the sticky end producing and blunt end producing restriction endonuclease. Right. So what are the different examples? Examples for sticky end producing restriction endonuclease will be ECO R1, TAC1, BAM H1, MST2. So these kind of restriction endonuclease which are isolated from your bacteria. Now there is one example that is given for blunt end producing restriction endonuclease, that is HPA1. Yes. So this is what is all about your restriction endonuclease. So what are these sticky ends? They cut in such a way that they produce hanging ends which are complementary to each other, and these ends readily pair. So how will it be useful? Because I'm cutting the human DNA of interest as well as my vector by the same restriction endonuclease, I will get the ends which are hanging from these two. Yes, I will get hanging ends or the sticky ends from the vector as well as from the human gene of interest. So these two, since they are hanging and they are complementary to each other, they can when they are put in the same medium, they can easily anneal itself, right? So thereby I can get a transformed DNA. Yes, I can get a transformed DNA or a recombinant DNA. So blunt ends are nothing but they cut the sequences in a straight way. So thereby it produces blunt ends, right? So this is about your restriction endonuclease. The second tool that you want is vectors. So what is the second tool that you need? Yes, the second tool that you want is vectors, right? So what is a vector? Vector is a molecule of DNA. Usually it is from your bacteria, right? So I will tell you, explain you how. But not just bacteria. We also have other organisms or other vectors or artificial vectors. So it is a molecular cloning vehicle. So what is a molecular cloning vehicle? Vehicle means it can carry something, right? So my vector is designed in such a way that it is carrying my gene of interest. Yes, it is carrying a gene of interest. So it is capable of carrying the gene of interest. So vector is a molecule of DNA to which the DNA fragment of interest is inserted and then cloned when it is transferred to a host cell. Right. So what are the properties of vector? So if I have to use a vector, there must be criteria that the vector should satisfy. The first and foremost criteria that a vector should satisfy is this capacity for autonomous replication in the host cell, which means that it should have its own ori, or it should have its own origin of replication, right? So it has the capacity for autonomous replication in the host cell. Okay.